So both Crohn's disease and ulcerative colitis, they're very heterogeneous diseases. So every person sort of presents slightly differently. So there is a lot of variability sort of within individuals. So this makes it very challenging to find a treatment that sort of fits everyone.
The challenge is to identify people who have a fairly aggressive course of disease and need more drug therapy or surgery, and differentiate those from people who have a less aggressive or more indolent, benign course of disease, and to give each one the correct therapy, balancing up the need for treatment against the safety considerations and other considerations involved in starting drugs.
Personalised medicine can really have a great impact for diseases such as Crohn's disease, and that's because we really want to be able to tailor certain treatments to each individual patient, and because of our DNA, we need to be able to incorporate this into our treatment decisions, and so this is why the METHYLOMIC project is really important.
On clinical grounds, even the best clinician in the world, the most experienced clinician in the world, using the currently available tools doesn't have much more chance of doing that than by flipping a coin. So personalised therapy is trying to move us on from flipping a coin and offering a drug which we're not sure whether will work, to actually having more confidence that we're offering a specific person drug A because we know they'll respond to drug A, and not offering them drug B because we think their likelihood of responding to B is less.
The hope is that the IBD field can sort of mimic what's been happening in the cancer field, and that's that we can have this personalised medicine approach for all patients with IBD.
I guess that's the goal of the trial we're talking about, really, to take things to that level. Having said that, you're right, in cancer treatment, molecular signatures are helpful, are used frequently, either in the blood or in the cancer itself. Often it's in the cancer itself that the molecular signature is more important. So that's the goal. We're not there, but this trial is maybe the first step along that pathway, or is hoped to be that.
So personalised treatment aims to be able to focus on each individual's sort of unique genetic makeup, and if we can find a way of being able to detect this, then we can choose what is the best treatment choice for each individual. And so currently there is no best decision for deciding what treatment to give a person. So looking into their own DNA will actually be able to help better suit these treatment choices.
We're born with this genetic code. You inherit one, as you know, one chromosome from the mother, one from the father, and together they make you the person you are. So that's the germline DNA, but actually methylation is and that is inherited, but the methylation are modifications to that code that you get from your mother and father that make modifications to the proteins and to the tissues of your body. So it's an acquired, or there's a lot of controversy as to whether the methylation can be inherited to some extent from mother and father or not, but it's a change that's on top of the DNA that's inherited from mother or father that can alter critically the tissue of the body. And the hypothesis that we have is that the methylation changes in people with inflammatory bowel disease, Crohn's disease and ulcerative colitis somehow firstly have given rise to that person developing the disease, and then for the purpose of this trial, the methylation signature also might be critically important in determining whether they'll respond to therapy.
So epigenetics is on top, and our genetics is our DNA, which is in every cell. And epigenetics, so these modifications that occur on top of the DNA, can actually change the way different genes in our body are expressed. So in some instances you may find epigenetic modifications, which actually causes a decrease in gene expression, and these genes can be quite important for disease, and so if we can find these epigenetic modifications, it might help to be able to target our treatments to these certain areas in our genomes. So I suppose we could say that epigenetics is similar to formatting text. So you can read text in many different ways. It could be in italics, it could be in bold, it could be underlined, and each of these different formats sort of reads very differently, and that's quite similar to epigenetics. It can change the way these words are said, and that sort of shows the way that our genes could be expressed. They're sort of read in slightly different styles. So lifestyle factors and even the environment can have quite a big impact on DNA methylation. So for instance, smoking can cause changes, also things like air pollution, but even lifestyle changes such as exercise or the things that we eat can also impact DNA methylation.
We hypothesize that it's the interaction of genes and environment that's involved in why someone gets inflammatory bowel disease. A proportion is due to the genes, the structure of the genes one inherits from the parents, and a proportion to the environment the child is brought up in. And it may be that environment affects the genes and gives rise to disease by causing methylation or demethylation of key areas of the genetic material. That was the hypothesis, and then we've gone on in a series of studies over the last 10 years to show that there are very specific changes in DNA methylation associated with Crohn's disease and ulcerative colitis.
So you could have two patients who present very, very similar to each other. However, they may respond very differently to different treatments, and that's because our genetics and our mutations that we all have can sort of prevent us sometimes from responding to certain treatments. And this is why personalised medicine is really, really important. So DNA methylation offers the potential to find biomarkers that can then be explored in more detail, and hopefully we would like to see that drugs are then made to target these biomarkers, and hope that we can sort of extend the current sort of biological therapies that are currently available for patients.
The OMICROHN clinical trial is really designed to validate a biomarker panel, and a biomarker is something you can measure in the blood of patients, so to show that a biomarker panel can predict which treatment is the best for patients with Crohn's disease. And in order to do so, we need to organize what we call a prospective clinical trial, meaning that there are two groups in the trial. One group of patients that receives random treatment, we call that standard of care really, and the other group of patients that receives treatment based on the biomarker that we measure in their blood. In order to validate, to show that the biomarker is really valuable, you cannot do research at a single hospital, because that would include the risk of bias. So you need to do this at multiple hospitals, and preferentially in multiple countries, because it could be that the biomarker is a little bit different in patients that live in Italy, for instance, or in Greece, versus in patients that live in the United Kingdom. So that is why you do multi-center and international trials. Also, it's very difficult to do trials of this magnitude with more than 300 patients in a single country. We need to do this in a timely fashion, so that's another reason to really include almost 50 clinical centers across 7 countries in Europe. The biomarker that we measure in the blood of patients is measured at the beginning, before the treatment, really at the beginning of the trial. But we also want to investigate if the biomarker changes over time. In other words, if the treatment itself changes the biomarker, or when the disease becomes quiescent or response to the treatment, that the biomarker changes. We also take biopsies in the patients because they undergo a colonoscopy in the beginning and 6 months later, and we collect biopsies to see if not only the endoscopy improves, in other words, if the ulcerations go away, but also if the inflammatory cells disappear from the inner layer of the bowel. Well, in order to demonstrate that a biomarker-guided treatment is of value, in other words, that it is better than standard treatment, you need to demonstrate that in one group of patients receiving the biomarker-guided treatment versus another group of patients that receives standard of care. And we aim to demonstrate that the biomarker treatment is at least 15% better, so 15% of the patients more in remission without ulcerations after 6 months. So we measured the methylation of the DNA of participating patients with the Illumina platform, and that measures 850,000 methylation sites in the DNA, okay? Now, you receive then data that is really impossible to understand because it's millions of data points. Now, that data goes into a computer algorithm, and the computer then decides based on all those data, and these are very complicated formulas that no human person can explain, but the computer generates an AI-developed, artificial intelligence-developed algorithm that then determines the ideal treatment for the individual patient. For patients that participate in the OMICROHN trial, there is virtually no risk because the two treatments that we investigate, that we administer, ustekinumab and vedolizumab, virtually have no side effects. So there is only potential benefit for patients to participate because either they receive the standard treatment, which is of low risk, or they receive a better personalized treatment, which also carries a low risk. So for patients, there is only a win-win situation. Well, the value of epigenetics to predict response to treatment is something that we established in earlier research. And just recently, we published a paper in The Lancet demonstrating that methylation is indeed a sensitive marker associated with response or non-response to specifically ustekinumab and vedolizumab. And currently, we're investigating in other cohorts of patients the same idea for infliximab and adalimumab, two earlier treatments, really older treatments that target TNF, so they have a completely different mechanism of action. But we are confident that the methylation will also predict response or non-response to those treatments. And eventually, we may even decide to bring those even older treatments also in the OMICROHN clinical trial. So in the future, if the OMICROHN trial is successful and really demonstrates the 15% superiority of the biomarker-guided treatment, epigenetic-guided treatment versus standard of care, I think then it might become standard for doctors to send out a blood sample of patients to a laboratory where the epigenetics is measured, then the algorithm is applied with artificial intelligence, and doctors and patients receive the result. Listen, this is the best treatment for you. Why don't you start this treatment better than other treatments that might not work? The value of epigenetics in treating and handling and following inflammatory bowel disease is just in its infancy. And we're even expanding to other disease areas like psoriasis or rheumatoid arthritis. So, and not only the response to treatment might be predicted in those patients, but also the development of complications like fistulas or joint pain or other problems, cancer maybe. So we're just at the beginning of that research, and in the future years we'll learn really a lot more than what we know today. Well, we are very grateful that the patient associations that is coordinated by IFCCA, the central, let's say, federation of the patient associations in Europe, that organization is supporting this clinical trial. We know that patients are very concerned about receiving the right treatment, about also avoiding complications of treatment, and we are very grateful that we can progress science in this direction So it's really a collaborative effort among patients and clinicians, and that's so much fun.
It would have made a big difference, huge difference, when the doctors right away knew which medication I need, because I think I still, yes, it would be a whole other life. I would be working, it would be more joyful, more light, more a bigger family, brothers and sisters for my daughter. I think I see a life with energy, energetic life. So yeah, it would be made a huge difference.